Welcome to Unlocking Science. Our goal is to glorify God by studying and unlocking the secrets of His amazing creation. I'm your host, Mr. P, and today we're going to be talking more about flowers, and I'm joined by Dr. Jennifer Rivera, and she's going to be here. She's got some fun stories to tell us about her time learning. So when we think about the idea of flowers in our episode called The Function of Flowers, we talked about how God created flowers. We read from Genesis 1 that they're created according to their kind, and that they have seed in them to produce after their kind. So have you ever planted spinach seeds and gotten dill?
No, I actually haven't planted much of anything because I don't have a green thumb. So,
Right? But if you have ever planted a garden, or you go to the store and you buy a little packet of seeds and you plant those pumpkin seeds, what do you expect to come out of the ground? Pumpkins.
A pumpkin.
Okay? And that's the way God created things. From an evolutionary perspective, why shouldn't another type of plant pop up from that, or some different types of variations over time? So we see a very discrete unit, a very clear unit of things inside the plant world that we can think about created according to their kinds. So roses are always going to produce more roses. Lilies are going to produce more lilies and types that look like them. And that's an incredibly important concept. Now, I haven't always believed that way. I used to be an evolutionist. Okay? I was trained in the evolutionary way of thinking when I was in college, and when I started teaching science, I taught it from an evolutionary perspective. But I didn't understand a lot of the issues involved in there, a lot of the assumptions. I just believed what my professors told me. I thought science was the way to know truth. And that was a little bit of a shift for me when I came to a biblical understanding. God changed my heart and He changed my mind on those issues. And now I understand things from that biblical perspective. And I understand why that is. I wrote papers about plant evolution in college and studied that in a lot of detail. But now I understand all of the false assumptions that were hidden inside of there. Have you ever had any experiences with that in the education world?
yes, and I definitely, with students as well who come in, and if you're from a Christian teacher and teaching from a biblical worldview, they have a lot of confusion, about things. But we can always go back to God's Word. We can trust God's Word. And God's Word actually says that, it opens our eyes to the truth. And you can only see the truth if you start with God's Word.
Yeah.
So, and it makes everything clear for us. So,
All right. So we're going to cut up part of God's beautiful creation, but we're not doing it just for no purpose. We want to try and understand how God has created things. So this lab we've got for you is a flower dissection. Now, we're going to look at the lily today, and lilies make a great specimen for us because they're very open structures. So if we think about this big yellow lily that I've got here, you can look inside of here and you can see all the different parts in there just with your eye, just peeking in there. You don't need any special tools to be able to see that. So if we think back to our episode, we talked about all of these different structures. So these outer pieces here are the calyx, and then we have the petals on the inside. We have the stigma here, the male parts, and then we have the pistil here, the female part. All those parts of this complete flower are clearly seen. Now this is a monocot. It's got petals in groups of 3. So we can think about this. This is a perfect flower also because it has both the male and the female parts. Those are all things we discussed in that episode. Here's a rose, and it's a different type of flower. You can see it has a different leaf pattern. This is actually a dicot, and it would form a different style of seed. It's got veins in its leaf that spread out in a web out from a middle vein here. Okay, a little bit different. If we go into here, we can see the sepals around the bottom that would form the calyx. We've got the corolla here, the petals, and all the parts inside are hidden. And we cultivate these types of roses to be more beautiful. Okay? So we can use God's creation and our knowledge of how we crossbreed these and how plants pollinate with one another to create these beautiful structures. But because the lily and the rose are 2 different kinds, are we ever going to get a lily rose to be produced?
never.
No. And that very fact of the pollen that comes from the lily is never going to fertilize those ovules that are inside of the rose. Why not?
It was kind of funny because it reminds me of a story. I was
Okay.
taking a botany class with my daughter, and the teacher was an evolutionist, but she was very knowledgeable, had a master's in botany, and she knew a lot of information about the structure of plants. But she was describing the different parts, which we're going to learn today when we dissect our flower. And she was talking about the stigma, which is the female part of the plant. And part of the stigma, the very top of it is the stigma. And the stigma is attached to the style, which is then attached to the ovary, which is down here. Now, that whole complete thing is called two different things, but we could say it's the pistil.
The pistil.
It has a couple different names.
Yep.
But the very top here is the stigma. And when the pollen falls on the stigma will allow those pollen granules to go down to the ovary. And this evolutionist instructor said, she goes, it's just interesting though that plants will only allow the pollen down from, plants that are within their same taxonomic family, right? A lily is not going to allow rose pollen down its stigma because it won't fertilize its ovary. There's no sense, it doing that.
It's not going to let them in if they can't do the job that
exactly.
it needs to be done.
And it's just evidence that
it's almost like God
plants
created things
after their
according to their kind,
right.
to reproduce after their kind.
That's exactly what we see.
So they can, from an evolutionary perspective, they can recognize those things, but they can't attribute it to God, and they can't give him worship and praise for the way he's designed things. Okay, so for a basic flower dissection, you don't need any fancy tools. If you maybe happen to have a dissection kit, if you use those for homeschool things, you might have things like these little probes and scissors and little forceps and maybe even a scalpel inside of there, those types of things. You can use something like that, but that's not necessary. You could use something as simple as a steak knife or a pocket knife, a pair of tweezers from the bathroom, those types of things. And if you don't have a pair of forceps this big, it'll be okay. You can just go with the smaller ones, all right? But I do need those every once in a while. So it doesn't take a lot of special tools. No. You might also want to have something like a hand lens or some type of magnifying tool. Sometimes you can
Microscope,
get apps for your apps for your phone that can turn your phone camera into a magnifier, basically. Microscope, if you have a microscope, you could do those. Now you're going to be looking at pretty large structures, so you'd have to use the lowest power setting and move the stage way down. But a microscope would be
it's really good
as well.
for the pollen, though. Yes.
Okay, so let's think about the basic structure of this lily, and how would somebody go about dissecting this lily? What kind of steps would you want to walk through
well, the first thing that you need to do is you need to, I would say, remove the flower itself, right? So you need to remove the flower.
Now you could use some kind of tray or some type of paper plate. It doesn't have to be anything special.
And the next thing we do is we want to get down to those male and female structures so that we can really see that. And as Mr. Patterson was saying, they're in multiples of 3, and what we actually have is 3 sepals and 3 petals. Together we can call those tepals. So this actually has 6 tepals, we would say, right? But it's very easy. You don't even need scissors for this. You can literally just gently
pull them down gently.
Just pull these down like that. Try not to lose your
I'll grab that one.
stamens, okay, which are the male parts of the plant. All right? And so once you have your petals on your tray, you want to take a look at your stamens. All right? And now we can really see, let me lay this down because it might be easier for you to see if I lay this down here.
Let's bring it out to the front here where the camera can get a little closer. All right. So here you can see they all connect down here. So this is where the petals and the sepals were torn off. And this structure here, this long filament, is called the filament. And it connects up here to this structure called the anther, which has the sporangia and microsporangia inside of it. And let's count them up. 1, 2, 3, 4, 5, 6. That might be a clue.
Multiple of 3.
It's a multiple of 3. If you're using a different type of flower that's a dicot, you would probably expect things to come in multiples of 4 or 5. Okay, so let's go ahead and just continue removing those carefully. You just kind of peel them down. They should just pop off. And then
what's all that orange stuff, Mr.
We'll
Patterson?
get there in a minute. Let's keep working here. We'll talk about that in just a second. Okay, so there are the 6 stamen pulled off. Now we have this structure. So you were talking a minute ago about the different parts of the pistil. This whole structure is called the pistil, and that's P-I-S-T-I-L. Up here, notice that this looks like it's the same color as all of this powder here. That's because these pollen grains that are in here have fallen on the surface of this, and it's sticky. and they've attached to there. So this surface is called the stigma. And then we move down here along the style, and then down here, you can see it gets a little bit fatter in this section. That section there is where the ovaries are. Okay? So there are chambers in there and little ovules. The pieces around it are called the carpel. Lots of other names we could use. But as you work through the activity, which you can download the PDF for this, you'll find the information for that down in the description of the video. Download that, it'll walk you through all these steps. Now, what would we find if we carefully maybe cut the ovaries here in half, or even up here at the top of the stigma? How many sections would we find in there?
It's kind of interesting because you have to cut it really thin. It's very difficult, like with a razor blade. You'd want your parent to help you do this, and you really need a microscope. It'd be very hard to see without it. But if you take a cross section of the ovary, you're actually going to see pods of 3 right inside of the ovary, which just points us back that it is a monocot, and you can also see lobes of 3 on the top of the stigma, which you actually might be able to see.
Yeah, let's see if we can get the camera down there and zoom in on that. You should be able to see 3 little heads, and one here, and one here. And each of those corresponds to the different ovaries that it's going to go down and connect to.
And we would say this is a superior ovary because it is above the peduncle there, the base of it.
Or hypogynous, if we want to get super fancy. Here is a structure that came from another type of lily. It's not this exact one, but this is actually the scar that's left over. So right here, if we lay these down next to one another, this little skinny structure here, that's the ovary structure, would actually be this same thing right here. And notice that this has 1, 2, 3 ridges on it. And this little triangular shape across the top is a scar where the top of this style, which is over here, has fallen off. So this piece would correspond to this, and then this is corresponding to this. So this is actually a ripening ovary, and we typically call that a fruit. Okay? So this is the fruit that a lily would make. Now, I've never had lily fruits before. I might try it tonight. I'll find out whether they're poisonous.
I don't know. I heard they're poisonous. Someone commented on our show they were poisonous.
Oh, okay. So we want to be
don't
really
ever eat
careful with
anything.
plants unless you are clearly researched it and know what they are, not to eat them. But if this were an edible fruit, that's the structure that we'd eat. Now on a rose, you do get that type of structure, and it forms down here at the base, and we call those rose hips. So if you see on a rose bush, after all the petals fall off, this little ball will form down here at the bottom, and that's called, we typically call those rose hips, and those are used for medicinal things and flavoring of things. So if we were to look inside the rose, we wouldn't find things in groups of 3s because it's not a monocot. We'd find them in groups of 5s or 4s for other dicots. So all of these different structures are really cool to look at and really cool to explore and understand how God created them. All right, so here's another little thing you can do, and this would be great if you had a microscope, and you could even do this with a hand lens. If I take this piece of paper right here, and I put one of these anthers that's got all of those pollens on it, can actually knock off some of those pieces, and all these little pollen grains, do you see that brownish stuff? All of those little pollen grains are actually the things that hold the male gametes, the sperm, to carry them down to the egg. And if I kind of smear this across here, you can see it makes kind of a, oh, we got yellow on yellow. That was a great job, Mr. P. But you can see it smearing out over here, and you see it all over my finger as well. You get that color, that very distinct color. So that's part of the pigment that's inside the pollen. So you can actually look at the individual pollen grains if you have a microscope or some way to magnify that a little bit. Other things that you could do to explore this, you could find more mature structures like this if you've got anything. You could cut this in different sections, see what's inside of it, see if you can identify where each of those little ovules is, how many chambers there are. Each of the seeds might be sectioned off into a specific little locule. They're all different kinds of structures. So if you go and explore some of these different things, you're going to find really cool stuff that God's made in different patterns. Other ways that you could explore, you could look at the seeds that are produced from them. the different colors that you find in the flowers, the way that things are arranged on the stem. There are just so many things to study about plants. They just have such variety in all the ways God's made them. Okay, so you're going to be doing this dissection of this lily or another flower that you can find, and we want you to be careful. Slow down, take your time. We've got some questions for you to think through, and especially some biblical application as we think about how God's made these plants and how we can glorify Him through all of those things. We hope you enjoy this dissection and learn all about God's amazing creation. And until next time, get out there and explore it.